So thank you, Siobhan and Colleen, of course. So it's an absolute honor to do this today. So also, I'm also speaking for Karin. So she will come up in 10, 15 minutes for the second part. And I want to start off by saying that I'm astonished. So I think we're not actually the experts. I think the patients are actually the experts. And I'm astounded by the moment you get the diagnosis of such a disease, how fast and quick you can obtain all this information. You have to absorb this, you have to deal with this. And I think there's an important role for medical doctors and clinicians to guide patients through their treatment, but there's also an important role for scientists as well. And I think that's a bit underwhelmed. So I feel that as fundamental scientists or discovery-based scientists, we sort of have to guide the patients through Google. Right? So there's so much information and there's so much knowledge with the patients that they just have a hard time to put this on a spot and how to deal with this. So Karin and I were discussing what we should do today because we wanted to be low-key, we wanted it to be interesting, and we wanted to be focused on the patient. And also yesterday we realized that a lot of stuff is already covered, so regarding the biology, but also in terms of the clinical aspects. So what we're going to do today is, so this will be very hard for me. So I'm trying to give a very short overview of all the years of lobular cancer research. And there are people that are way more experienced with this, and Matthias is here, so interrupt me anytime, right? And anywho, we feel as well that this should be sort of informal, so just feel free, people with more experience, so interrupt me if I forget something, okay? So after this part, Karin will come up and she will discuss the leaflet and the clinical aspect. Okay. Oh, wait. So let's go. So yeah, the first thing that you then do, you go on PubMed, and I just typed in breast because I was curious what was the first official breast cancer paper that was published. And the first paper I actually found was from 1720, and I thought, okay, now we're going in with a bang, with beautiful drawings, etc. But it was actually a male student who swallowed a breast pin by putting on a dress. That was the first official paper regarding to the breast. And the very first real paper, also scientific paper, so to say, was in 1783, where they described how to do a mastectomy of the breast on both sides. And actually the first official review was almost 150 years later, in 1916, where they discuss, they go a bit more into details in how the breast looks from the inside and how to deal and to clean the tools very well and how you prevent infections, etc. And it was until 1920 that the first paper popped up where they sort of tried to describe lobular breast cancer, but they did not really have a name for it. So they called it papillomata of the breast. So there were structures within at some point that they could not really type, so what it actually is. And so this was discussed yesterday as well. So 20 years later by Foot and Stewart, and this is actually the first histology slide image from an invasive lobular breast cancer, and these two pathologists were the first who actually presented lobular breast cancer. So then it went on, and it took roughly 20 years before the first mammogram was published of lobular breast cancer. And what I liked a lot is that, so although this was 20 years later, they had sent all the images to the pathologist, so Dr. Foot, to confirm if it was actually lobular breast cancer, yes or no. So these were the two, so these were the pathologists who described it the first in the previous slide, what I showed you. And so they checked and cross-referenced with them all the slides if it was lobular breast cancer, yes or no. And it was. And I think, so at that point, there's just many years and years of case reports. They find lobular cancer there, they find it here, but they not really understand the disease. There was a lot of debate about the biologic nature, and also going through these articles is also quite interesting how they write it up. So they use the actual names in the articles, like Sir Henry III thought this, blah, 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 etc. And there was a lot of stuff regarding diagnostic features and therapeutic management. And I think there was a hallmark discovery in 1977, where for the first time they did a serial transplantation in mice, and they found that breast cancer growth can be hormone dependent. And this was a major breakthrough. And it only took 2 years by Anatoniades, and that they actually found that in general, the lobular carcinomas are hormone positive. Right? So this was a big jump in the field, so to say. And I felt through the articles that the pathologists they needed something, they needed tools to better cell type the malignancies that they were seeing. So they wanted to understand the tumor better. And this is, which I think was an important step, that they sort of discovered the keratins. And they were able to distinguish luminal cells from myoepithelial cells. So they were able to better discriminate the cells in the mammary gland. And this is I think one of the first IF images of that. No, it's not really IF. I'm not sure now. But they took lysates from a rat. So these were immunized rats. And as you can appreciate here, so here you see luminal cells, and here you don't. So back then they didn't know, but they sort of did the first luminal staining. Okay. So then in the 80s, we're sort of ramping into the age of the molecular biology. And from this point on, a lot was happening. So scientists were really searching for the genes that would cause healthy cells to transform in tumor cells. And in 1984, both oncogenes and tumor suppressors were first described by seminal articles. And the most prominent are still heavily being studied today. So MYC, RAS, the ERBs. So ERBs are the HER proteins, P53 and RB. So all these proteins, they're still heavily under investigation. And these are two landmark papers from the Weinberg lab, where they discovered actually HER2, and from the Papadimitrou lab, where they discovered P53. And I think as a scientist, doing research in this age, it was really exciting, because this is like, so I always like to say these are the dinosaurs of the molecular biology. This is where they found how cells got transformed. And I think, so this also started the stage of molecular phenotyping tumors, right? So what oncogenes and tumor suppressors are expressed or not. And they found, and I think it was also an age where they just stained everything with everything. So, okay, if we know these proteins, we have to stain everything that we got and see what's positive or not. Although not fully understanding and grasping the biology. So they found, for instance, that in lobular carcinoma that they have high MYC expression. So I'm not sure if this still holds today. So this was in 1987. In general, that they had low HER2 expression, and I show an image of that here. And that they are also in general high in RAS expression. And I think in the beginning of the 90s, where scientists were more like trying to investigate the drivers of invasion, pathologists were more searching for the markers of invasion. And in the 90s, what you saw more and more is that in the past years before that, more and more cell lines were isolated. So scientists learned how to isolate cells from patients and culture them actually in addition in the lab. And one very classical ILC cell line that we all know is SUM44PE, and we still use that today. It's sort of our, I think it's almost safe to say that's our benchmark ILC cell line to do certain investigations. And if you go back to the old papers, they do note that there are floating cells, but they don't understand it and why it's happening. And I think that was an interesting sentence. And it was in 1991 where the Birkmeyer lab discovered that epithelial cadherin, so E-cadherin, can be linked to the invasive and the non-invasive state of breast cancer cells. So this is the first IF I could find on breast cancer cells of E-cadherin. And on top we have the MCF7 cell line, which we still use today. So actually MCF stands for the Michigan Cancer Foundation. So this was a time where labs were trying to create cell lines, and they gave it the name of their own institute to sort of claim it. So like, see what we did. So this is MCF7, and MB43, which is an ECAD negative cell line. And here Burgmeier also describes that ecadherin is the adhesive glue that sticks the cells together. So what was the molecular driver of lobular carcinoma? So there are quite some papers of this, and scientists were working very hard to get this question answered. And in 1993, they did an ecadherin staining, and they found that 85 of ILCs had loss of ecadherin. And it was in that same period that they found that for classical ecadherins, that a certain chromosomal arm, so they were karyotyping tumors back then, that it was lost. And on this chromosomal arm are basically all the cadherins. So ecadherin, p-cadherin, and there's also k-cadherin, r-cadherin. And they're all on the same chromosomal arm, and they noticed that lobular carcinomas lack this chromosome, or at least it was unstable. And it was in 1995 that it was clear that there was a causal link between reduced or lost ecadherin and lobular carcinoma. So molecularly, they studied this way more, and they did way more advanced experiments, and around the 2000s, I would say, it was just done and dusted. And as well, at that point, they say that there was a negative correlation with HER2 when ecadherin was lost, and that there was a positive correlation with hormone receptors. And we know by now, of course, that this is not entirely true anymore. And then sort of the age of mouse models began, molecular biology, and also genomics. And I think now there was sort of a period where, okay, we know all the oncogenes, the tumor suppressors, we know sort of what they do in which type of cancer, but they also found that it's just not one mutation that can drive this disease, but it can be multiple mutations or multiple losses. And so scientists became interested, so what would be the evolutionary hierarchy of mutations within a tumor, as well for lobular carcinoma? And to study this, more cell lines were generated, so we entered also an age of molecular cloning, so we could efficiently put genes into plasmids, in vectors, which are like short circular DNA strands that we can put into cells and then express our gene of interest. And more cell lines were generated and published. And so I did really my best to not show Patrick's paper. You have to believe me. But he was the first, I'm sorry, where he inactivated ecadherin in combination with p53 and found that this was, in mice, leads to lobular carcinoma. And another important observation confirmed by Steffi, or the other way around, I don't want to get into this debate, that when cells have ecadherin and you put them in suspension, so you put them sort of in a bathtub and the cells are floating, they survive. So they are able to survive cell death. So they become anoicus resistant. And by now, we also understood why all these cells were floating if you put them in a dish, because they do not have ecadherin. And this gave rise to the first mouse ILC cells, which were widely distributed and could easily be studied in the lab. But also genetics came in, and this was also like the time point where pathologists and scientists found out that there is molecularly more going on in breast cancer, and that we can actually utilize the expression of molecular markers and thereby categorize breast tumors. So this is an image of a targeted cDNA microarray. So what we couldn't do back then is just do a whole genome on the fly, but you have to sort of pick and choose your genes of interest, and then you can see if they were expressed, yes or no. And this is actually the first cDNA microarray where they found that you have HER2 amplified tumors. And a few years later, it was actually also found that by doing targeted sequencing, that within ILC, there is there's a burden of PI3K mutations. And if you go into the percentages, they differ a lot around this time. So there are papers that claimed around 30%, but there were also papers that claimed around 60%. So, and I think that was more due to the effectiveness of the sequence arrays at that point. And we're getting closer to now, and I think this was a hallmark story as well, in where they did, I think it's now, this is sort of benchmarked the whole way we molecularly now subtype breast cancer. So they did a massive experiment by completely categorizing all the genes that are expressed, yes or no, in all breast cancers. And they also looked at the active or the inactive state of proteins. And also here they say, they do for the first time, they categorize not ILC as just being luminal A, but as a separate entity within the molecular subtyping of breast cancer. And here already they found that there is an increase in the activation of AKT, and also that there is a loss of PTEN, etc., which is different from other luminal A hormone-positive type of breast cancers. So what also got published was the first overall survival study of lobular breast cancer compared to ductal breast cancer. And what I actually like a lot from this graph is the biology behind it, because I feel this is one of the first studies that actually shows that ILC has long latency relapses, and that there's actually a period within the progression of the disease that we have cells lingering in the body that in the end can reawake and be invasive. So as a molecular biologist, so this opens, yeah, like a whole toolbox of idea of things that we can tackle and what we should study to prevent this from happening. Okay? So we're entering the CRISPR age, more advanced cloning and the culturing of organoids, and do PDO, PDX in mice. So CRISPR and advanced cloning allowed the generation of more ecadherin mutant cellular models. So we can now quite easily either remove ecadherin from cell lines or re-express. We can do immunofluorescence on them, and we can combine oncogenes and tumor suppressors and study them in mice. And what we now also see more is that ILC tumors, so this has been discussed extensively yesterday, can be isolated from patients and cultured in 3D, and we can also genetically alter them. So here you see examples of organoid models in 3D. So this is actually a result from our lab where we CRISPRed a residual ecadherin molecule which was mutated out of a patient model, and we can also grow them in mice. So where does this take us now from the biology point of view? And I think the coming 10, 15 years will be quite exciting. So from the signaling point of view, so this is more like my personal opinion, right? So this is not set in stone whatsoever. I feel that we should better understand the HER2 and HER3 mutation, and then in relation to AKT and PI3K signaling, we now have these beautiful tools where we can really zoom into the cellular architecture of a cell and try to understand how these molecular cues are regulated and how these are communicated. So the consequences and co-occurrence of these mutations and how relapse works on the activation level and based on the targets that it's signaling through. The ILC tumor microenvironment, right? So we have seen in multiple studies that it's different compared to an IDC. So the role of CAFs, cancer-associated fibroblasts, growth factors within a microenvironment, the composition of the niche from the primary tumor, but also from the metastatic site. The immunoresponse, so the body's response to an ILC tumor. So what is the composition of an immunoresponse towards a primary or a metastatic ILC tumor, and what is its niche? And I feel that we're also going to enter an age where we're going to treat context more instead of just a mutation. So the people that visited my poster, I already talked about this. So what we have seen in the past 10, 20 years or so, a lot of companies are like massively went on, okay, these molecules are mutated, we have to make inhibitors for these. So they work, but they're not curing. And what we see now, and it sounds a bit counterintuitive, that if we combine targeting a mutated protein with another protein which is not mutated but might facilitate its signaling, that this would benefit the treatment. So that we look more into the feature of the cells, where it's actually residing, and can we treat, for instance, this indolent, lazy behavior of ILC cells. Yeah, combination therapy, and maybe even immunotherapy in the future. So one more thing, and then I will stop. So what I noticed in these 80 years of ILC research, so 10% of the published work was immunology, biomarkers, histochemistry, and treatment opportunities. Roughly 5 to 7% of the publications are sequencing and omics, and only 3 to 5% is actual biology. So it's not a lot. And now we roughly still have 80% left. So this is actually what you find in every paper, and it's been stated yesterday a lot. And I think about 80% of all the published work is, with all due respect, Matthias, it's pathologists arguing. So what type of disease is ILC? And I think if just Foot and Stewart didn't call it a rare form, but a new form, it might maybe even have fixed a lot of arguing over those years. Yeah, so, okay. Karin?
Thank you very much, Thijs, for this very nice overview of the history of ILC biology. I will now focus a bit more on the clinical aspects of lobular breast cancer, the present, not the entire history, and some advice that we give for current clinical practice. I will do this by the use of our leaflet. I hope you all saw it. I hope you all have a copy with you. If not, they're still available. Please take one home with you. I will follow the structure of the leaflet throughout this presentation. A lot of what I will say is already mentioned yesterday in Summerschool and in all the great talks that we had. But it's just the idea to give you a bit of insight on how the leaflet is built up and how you can use it to explain to other patients, to family members, etc. So as I said, I will follow the outline. I will start with what is lobular breast cancer and focus on the main differences between lobular and non-lobular breast cancer. I will talk about the treatment in early setting, both local and systemic. I will talk about the treatment in metastatic setting, and we'll talk about the hereditary risk. In the leaflet, you also found the key concepts and additional information. This will be presented throughout the presentation, so I didn't make a separate topic out of that. So first, what is lobular breast cancer, and what are the main differences? I'm very happy that I can use this slide of Professor Flores, that the images, although it's not very clear, they say it all. So lobular breast cancer represents 15% of all breast cancers. And it was Professor Azzopardi who said that invasive lobular carcinoma are cells that are next to each other but do not touch each other. Here it's visualized by the use of marbles, so they're next to each other but they're not connected to each other. While invasive carcinoma of no special type, the cells do touch each other. It's here by the building blocks, they're really connected to each other. So I think it's a very nice visualization of what is going on. Then the main differences between lobular and non-lobular breast cancer. First of all, in the early setting. So early means that we only know that the cancer cells are present in the breast and maybe the axillary lymph nodes, but there's no evidence that there are already lesions throughout the body. So we know that for lobular breast cancer there are more often multiple lesions in one breast, that it's also less clear to see on standard mammography, so it's often underestimated, it can be even missed, and so we advise MRI of the breast in almost every patient with lobular breast cancer. Mammography can be enhanced by 3D techniques and contrast, but still we do advise an MRI. Also, ultrasound is complementary to mammography in the same way as it is for non-special type breast cancer. For the future, we envision new techniques for screening and diagnosis. Long-term benefits and side effects of repeated MRI need to be investigated further, and also the impact of breast density. At the pathological side, we already had a nice explanation from Thijs. So we know that they're not touching each other, that they have a larger tumor size at diagnosis and more nodal involvement. The majority has, as we saw yesterday, is of moderate grade of differentiation, but as we saw, it's not really predicting the prognosis of lobular breast cancer because it's not really applicable for lobular breast cancer, as was explained yesterday. So more than 90% of lobular breast cancer is sensitive to the female hormones, and only 3 to 13% of them express a protein called HER2, which can also be targeted by specific treatments. And so also there we need a clear guideline actually for the pathologist to improve the pathological diagnosis. We need improved criteria. Then moving on to the metastatic part, so the cancer cells have spread throughout the body and have formed new, not really masses, because lobular breast cancer is not growing into masses, but they have grown in other organs. And so as we already saw in several talks yesterday, they also metastasize frequently to the bone and to the liver. But for lobular breast cancer, the metastatic spread is more wide. They have more frequent metastasis to the abdominal cavity, to the stomach, to the intestines, also to the reproductive organs like the uterus and the ovaries, and they have less frequent metastasis to the lung. So the inactive state of those lobular breast cancer cells, since they do relapse later, that's still something that needs to be investigated more. Also, the mechanisms of metastatic spread need to be investigated more. As for the imaging part at the metastatic setting, the standard exams include CT and bone scan. It's both for non-special type breast cancer and lobular breast cancer. As we also saw yesterday, the normal PET-CT is less sensitive for lobular metastasis, but there are new modalities of PET-CT that are upcoming and that are under investigation. And I also want to emphasize that we also believe in the use of whole body MRI. Within our institution, it's already used a lot, and in my clinical experience, it is also much better than PET-CT to find metastatic spread of lobular breast cancer. Moving on to treatment. So again, the early setting, no signs of metastasis in other organs. We have two types of local treatment that can be performed. So local treatment is actually for the breast and for the axilla. So you know that the cancer cells are there, and you're gonna make sure that you eradicate them in the breast, in the axilla, by use of surgery. That can be either breast conserving surgery, where only the tumor is taken out with a bit of margin, or you can do a total amputation of the breast. For the axilla, you can either remove the first draining lymph nodes if you have no clinical evidence that those lymph nodes are affected, or if you have evidence that those lymph nodes are affected, you can remove the entire lymph nodes of the axilla. After breast conserving surgery, you always need radiotherapy to make sure that it's as safe as it is for mastectomy as it is for an amputation. But after amputation, there are some criteria also where you do need additional radiotherapy for the thoracic wall. And also for the lymph nodes, there are certain criteria to follow if you need to irradiate the lymph nodes as well. Then moving on to the systemic treatment, that is actually for the entire body. As I already said, so there's no evidence that there are any lesions in the rest of the body, in the other organs, but still you're not sure if there's already some cells that have parted to other organs, and so you want to reduce the risk that they ever grow out to form new lesions. That can be done by use of anti-hormonal therapy, also called endocrine treatment. that it's really important for lobular breast cancer since more than 90% is sensitive to those hormones. There are two main forms of anti-hormonal treatments. Either they go into competition with your own female hormones, that's called tamoxifen, or you have the aromatase inhibitors that decrease the amount of female hormones in the body. And I agree with Otto Metzger, as he said, that aromatase inhibitors are preferred in women that are postmenopausal. Then chemotherapy, we've heard a lot about it yesterday, in that it's common to think that it's less effective in patients with lobular breast cancer, but that's an open research question at the moment. There are patients that can benefit from chemotherapy, but it's up to the multidisciplinary board to decide if it's really necessary for each single patient. And then the targeted therapy, that's targeted against the HER2, that protein that can be expressed by cancer cells. That's also a form of systemic treatment. So every patient is unique, and also every treatment plan has to be unique, because also every breast cancer is unique. So it depends on the tumor characteristics, the patient characteristics, and also the patient preferences that lead to an individualized treatment plan for every single patient. So you can't just compare one treatment to another. And then moving on to the metastatic setting. There, the cancer cells have spread out through the body. So there it is important to have a treatment that also affects the entire body. So systemic treatment is the most important. It's not the aim to cure anymore. We hope in the future that at some point we can cure metastatic cancer, but at this time it's just to control the growth of the cancer as long as possible, to make it a chronic disease. Also there's no fixed sequence. It's a unique treatment plan for each patient. Although when the cancer is hormone sensitive, the anti-hormonal regimens are often given first. Chemotherapy is reserved for later lines unless it's not a hormonal sensitive cancer, or if there are any life-threatening problems. Chemotherapy works faster than anti-hormonal therapy, so then chemotherapy is given first too. And in some cases, we can also add the targeted therapy, so the anti-HER2 treatment. Clinical trials, so studies that compare one drug to another, can offer additional treatment options for some patients, but they have many criteria for patients to be included. So it depends a bit if you're eligible to participate in a trial or not. That's also a decision of the multidisciplinary board. And so local treatment is also used in this setting, but it's only for local control of metastasis. So it's used, for example, for pain relief, for bone metastasis that are giving some pain. Then you can irradiate those bone metastases, and that can help. Then, at last, the hereditary risk. We already learned a lot about this by the talk of Eleanor yesterday. So it is rare in patients with lobular breast cancer, but it's more likely in patients that are young, in patients that have lobular breast cancer in both breasts, and in patients with a strong family history. And clinicians need to make sure that they include gastric cancer for these patients in their family history anamnesis. As we saw yesterday, the risk for BRCA2, ATM, CHEK2, PALB2 is the same for non-special type breast cancer and lobular breast cancer. CDH mutations, and not mutations in the tumor, but in every single cell of the body, is more frequent for patients with lobular breast cancer. And BRCA1 mutations is actually only associated with an increased risk for non-special type breast cancer and not for lobular breast cancer. All this information can be found in the leaflet. It's now available in 10 languages on our website, so feel free to check out. Feel free to advertise this as well. And if your language is not up there and you want to contribute, you can always contact Thijs or me. We can provide you with a text that needs to be translated. It's just that we do ask that a researcher or a clinician is involved. Patient advocates may translate the leaflet, but there has to be a check from a clinician or a researcher. And then I would like to thank everyone who has contributed to the making of the English leaflet. So our patient advocates from ELBCC, also Christine, Patrick, Anne, Thijs, and also the medical doctors of UZ Leuven. And then, of course, everyone who has translated the leaflet into their own language.